days until break. Mr. D, any announcements?
Yes, I believe there. Wait, aren't your general public announcements?
No.
No, there are not. No, then. No, then I don't have any. Yeah.
Okay, well then I guess we'll go ahead and, with our presenter today. Sharanya Pulapura is presenting today on coronary stent fractures and how third-time presenter, so that means you can ask her any questions you want. You can like really ram her with the hard questions. She's ready for them. She's presenting on coronary stent fractures and the use of computers to do the testing on them, which is really, really cool. So with that, Sharanya.
All right, hi everyone. So my article is called Influence of Plaque Calcifications on Coronary Stent Fracture, a Numerical Fatigue Life Analysis, including Cardiac Wall. Or, oh, here.
In the meantime, Cindy's gonna sing.
No.
Come on, Cindy.
Or, why stents break? So, I'm going to start with a question. What do you guys think the ultimate purpose of medical research is? Anyone?
Make us live longer.
Yeah, I think we can all pretty much agree that the ultimate goal of medical research is to make people live longer. Problem is, the way research exists right now, it costs a lot of money, takes a lot of time, and it puts human and animal lives at risk. So what if we could do that in a fraction of the time, for a fraction of the cost, with no risk to anyone's lives? Now we can. It might seem a little bit far-fetched at the time that we could actually test medical devices and different therapies on the computer. But it turns out it's actually being used right now in the most deadly disease worldwide. Coronary artery disease. Like I said, it's the most common cause of death worldwide, and it's caused by a buildup of plaque in your arteries, which prevents blood from flowing into your heart. And the easiest type of treatment that can be given to people who are suffering from this disease is angioplasty and stenting, which is essentially you take a wire and you stick it up the person's artery. It's got a little balloon at the end, and the balloon blows up a small piece of metal called a stent, and that just holds the artery open to prevent the plaque from blocking the blood to flow to the heart. However, stents do have a few problems with them, the most common of which is stent fracture, which is just when the stent breaks inside of the vessel, which causes a series of complications, including in-stent restenosis, which is the most common one. That's basically when the blood vessel just fills right back up with plaque, even though there's a stent in there. Thrombosis, which is a blood clot, and even just sudden death. So obviously, this is a problem we need to deal with. There are two types of ways that a stent can fracture: static fracture and fatigue fracture. You could sort of compare it to like tearing a sheet of paper. A static fracture would just be taking a piece of paper and ripping it in half, while a fatigue fracture would be bending it back and forth until it eventually falls apart. So this paper is looking more at fatigue fracture, which is definitely the more common issue. And the ultimate goal of the study is to come up with an accurate computer model that can predict whether a stent is going to fracture under given conditions.
What kind of effects would a fatigue fracture, how would that be manifested on so what kind of damage would it cause?
Essentially, a stent has lots of little ring structures, like you can see over here. They're called struts, and they're like only a few microns long. So essentially they just snap. So sometimes it causes an immune response, sometimes it'll just puncture the vessel, a whole bunch of different things that can possibly happen. Anyone else? Or
What are stents made out of?
they can be made out of a bunch of different things. The most common ones are made out of cobalt chromium alloy, but there are some new ones made out of nitinol, which are it's like a shape memory alloy, and there are cheaper ones that are made out of stainless steel. Those are the golden ones. Anyone else? Okay, so the purpose of this particular study was to determine the effects of two different factors on creating an accurate computer model of stent fracture. The first is plaque calcifications. Like I said before, coronary artery disease results when you have plaque buildup in the arteries. So a calcified plaque would be a plaque with calcium deposits in it, essentially changes the makeup of the plaque and makes it a little bit more brittle. And the second factor is the effect of the cardiac wall movement, essentially just the outside wall of the heart and how it moves. And in particular, in comparison to pulsatile blood flow through the artery, because previous computer models generally factor in the blood flow, but not the motion of the cardiac wall. So it's trying to see if the motion of the cardiac wall is another important mechanical factor in creating an accurate model of the physiological conditions you find stent. So is it fun?
What causes plaque buildup in arteries?
I mean, sometimes it's genetic. Sometimes it's like dietary habits, like it's commonly found in people with obesity. Yeah.
What? Why'd you look at me?
Obesity is your deal, right?
Oh. I got
Is
nothing.
that good enough?
Yeah, I think so.
Good? Okay. Anyone else? All right.
Oh, sorry.
This is supposed to be the computer's presentation, too.
Fix it, Mark.
Okay. So, how do they actually make this model? I tried to simplify this, but feel free to ask questions if you don't understand anything. So essentially, to run the simulation on the computer, they need to make a model of the stent to actually do the tests on. So they use, in this case, they use an idealized 3D model based on the design of just a commercially available stent. And they use a technique called finite element analysis, which is one of the most commonly used, like even in like mechanical engineering. It's a very commonly used technique. So essentially what you do is you take the stent and you cut it up into lots and lots of really small pieces, and you're trying to solve for different variables such as the stress and the strain and other things that can contribute to fracture at all the different points along the stent. Basically why you're doing that is it's a mathematical equation that can't be solved unless you use guess and check. So basically they solve the equation at this particular value and this particular value, all these different nodes of the mesh, to ultimately come up with all these different mechanical factors that they couldn't just solve with math. Everyone good on that? Okay, so how they simulated the calcifications using this particular model, they varied the stiffness and the distribution of stress, which accurately represented the qualities of the calcified plaques. And they simulated the cardiac wall movement using a cyclic stress, essentially. As the heart moves back and forth, it's going to apply force on the stent. So they sped that up to, like, I think it's something that takes over the course of several years into just a few hours, which is, I think, pretty cool.
How did they determine how calcifications affect the stiffness and stress distribution in vivo?
Okay, I mean, the material, are you familiar with Poisson's ratio and Young's modulus?
Somewhat.
I mean, if anyone knows, it's Andrew, but...
Heard the terms, but go on.
So it's essentially, like, they're measures of that particular substance, like
All right.
essentially it's like stiffness / elasticity, and how the axial versus the transverse strains, the ratio between those two. So that's essentially how they simulate.
In fairness, Andrew, I asked her specifically to take that out of the presentation.
Okay. All right. So what did they find? First, we'll look at the calcifications. They found that the stress on the stent was slightly increased in the calcified regions in comparison to the non-calcified regions. So, like if you look at this contour plot over here, it's in that one over there. The greatest stress that the stent underwent was 798 megapascals, while in the calcified one it was 819 megapascals.
What are we looking at?
This is just a zoom in of the stent. It's a contour plot of the stress.
Of the surface?
Yeah,
Inner surface, outer surface?
outer surface. So basically, it's where purple is the least stress and red is the greatest stress. That's essentially what it's showing here. Okay.
So that's a surface or a cross section? So like, what's the gray line?
That's
The
just-
thick gray border.
Oh, that's. I guess it's just the background because this is the stem. That's just like if you looked at the outside of the stem.
And zoom in on the mesh, right?
Yeah, that's
Yeah.
Yep, that's
The gray perhaps vessel wall?
Doesn't matter, okay.
All right, we good so far? Okay. Gonna explain this. This type of graph it's called a Goodman analysis. And essentially all you need to know about this is everything above the red line is at high risk for a fatigue fracture, and everything below the line is at a lower risk of fatigue fracture. So the blue dots represent all those little points along the length of the stent which do not have calcifications, and the green dots are the ones that do. And as you can see, the green dots tend to be closer to the line or even above the line, while the majority of the blue dots are significantly below the line. So from this, they were able to judge that calcified regions are at a greater risk of stent fracture than non-calcified regions. Pay attention to this, we're coming back to it.
Over how long would we expect these stents to fracture, or what kind of time period are we talking about? Is it like months, days, years?
I mean, honestly, it depends. I think for the most part, it's years, but it can have like the sudden death or even the blood clotting. That's something that can happen over the course of days. But like the restenosis or like that tends to be more down the line. Okay. So in terms of the cardiac wall movement, this is actually where they saw the most significant results. That actually the motion of the cardiac wall had a much greater effect on the fatigue life than the blood flow did, which is interesting because previously the studies tend to only account for the blood flow and not for the motion of the cardiac wall. So just to give you some numbers, the highest stress in the caused by the stent only accounting for blood flow was 35 megapascals, while with the cardiac wall movement, it went all the way up to 390 megapascals, which is more than 10 times as much. Obviously, that's pretty significant. So here's another Goodman analysis. So I'll tell you, red line, same thing. The black dots represent the nodes with only the blood pressure, and the gray dots represent the nodes with the cardiac wall movement. Can anyone explain this graph to me now?
Not you.
This is like bio class. Andrew's the only one who ever knows the answer. Yeah.
So is it saying that the black part, when you're accounting only for the blood flow, it's not putting you over the red line, so it's not at risk for the stent to break, but with the pressure taking into account, then there are points where it is at risk?
Yeah, exactly. Thanks, Andrew.
Sorry. Does the gray account for both cardiac wall movement and blood pressure, or just cardiac wall?
So that was like a simulation where both of them were included. Sorry.
With that data, why weren't they testing the cardiac wall movement beforehand? Was it just like they
didn't have the technology, or was it?
I mean, they could have, they just didn't, I guess. But I think at the end of the day, the goal is to have a model which accounts for as many different factors as possible. So they figured, why not make one that can account for both blood flow and the cardiac wall movement to begin with.
How much of an effect does cardiac wall movement have, like alone, without blood flow?
Not actually done in this paper, but I mean, I could hypothesize that if you subtracted the black region from the gray region, that's what you get. But I'm not sure, so I can get back to you on that. I'll look it up. Anyone else? So where does that take us now? Future research. So now that we have a model which accounts for 2 new factors that can possibly contribute to stent fracture, we probably want to check that with mechanical benchtop testing. So essentially what that would be is you have a machine that can apply similar types of, similar levels of stress that we're expecting based on the models to see if the stents will actually fracture under those conditions. If they do, we can use that to verify that our simulations are working properly. We could also, like I said before, we used, in this particular study, there was an idealized stent geometry based on a commercial model, but we could also use a technique called optical coherence tomography to account for realistic stent geometries, and that's what Jacqueline and I are working on our IRT project. So essentially how that works is you take little slices of the stent, like pictures of the stent along the length of it, and you take all these little picture slices and you reconstruct them on the computer, so you get an accurate representation of an actual stent rather than just an idealized geometry. Eventually, we will have to compare the computer modeling to the in vivo testing, just to make sure that the results that the computer is getting match up with what's happening in real life, so we know that we can actually use the computers to replace the in vivo models. So eventually, at the end of the day, we're trying to create a better mechanism of screening stents for a particular person. So essentially, before we give a person a stent, if they have a stenosed artery, we could do a simulation under the specific conditions of their own body and the specific conditions of the stent that we're planning on using to make sure it's going to work before we actually give it to them. Akash?
So like right now, with like as you're doing like this on the computer, like is that just like a standard artery or like
Yeah.
is it like
Yeah, it's just like a standard, like an idealized artery. It's not like a specific one from a specific person at this point. So with the success of this study, we're hoping to see that maybe more diseases and more therapies can possibly be simulated on the computer so we can eventually get back to our main goals of saving people's lives in less time for less money.
Any other cases in which your simulation would be better than in vivo testing? I mean, like, you can't very well test chemo drugs on computer.
Oh, but you can. Don't you remember Smart Team? You can test, you can use
Okay.
MD
Yeah.
simulations to test that.
True.
How did they determine the, sorry,
Okay, we're rapidly reaching your question limit,
I
Andrew.
think I asked it a while ago. How did they determine how much the force was exerted on the stents by the wall of the artery?
I mean, they
Is that just based on?
they have like physical measures of how much force the artery exerts, and they essentially use like a cyclic like repetition of that particular force to see if that would increase overall the fatigue likelihood.
Last question, I promise.
So this is a relatively new form of, like a new way to try this out, right?
I mean, the particular technique of like finite element analysis is not new to mechanical engineering, but the fact that it's being applied to like more biological applications, that's what's even interesting about it.
And how do people know how accurate? Because they haven't done any in vivo testing, and So is this just like they're guessing that it's close, but
I mean, like that's kind of what we need to do to verify the models, is to compare it to what we're getting in Geneva. So essentially, like, I mean, if you want to know more, you can talk to me and Jackson. Like, because basically what we're doing is we're comparing our simulations to what MIT is getting in rabbits. So to verify the models.
how close are they to rabbits?
I mean, it's not published yet. We don't have anything conclusive. So get back to you when that's actually been done.
There's kind of a really interesting trust issue that works with computers and human lives, right? So the first kind of example was with insulin pumps for diabetics. And there was like a stigma that a diabetic could not really go to sleep with the insulin pump, the computer insulin pump, because if at any point the programming failed, the person would die, right? So there's always just kind of been this really interesting trust issue with computer simulations in biomedical devices, which is kind of a cool thing.
Are the manufacturers of the stents that are being used currently doing any of this benchtop testing?
I mean, obviously they do like physical testing and they do like clinical trials and that kind of thing. But this paper, the authors of this paper were actually collaborating with the FDA, trying to come up with a better way of screening stents, because the stents that have been tested are still failing even though they don't expect them to.